Hello, welcome to the second module of this third week of our course on Introduction to Dynamical Models in Biology. In the last module, we have introduced the basic concept of bifurcation. And in this video, in this module, we will discuss some application of those concepts of bifurcation to understand certain biological phenomena. Concept of bifurcation can be used to understand different phenomena at different levels of biology. For example, it can be used to understand population biology. It can also be used to understand molecular process phenomena involving molecules inside a cell. So at cell level also, I may apply bifurcation concepts. In this module, I will focus more on cellular processes rather than as a whole population behavior or ecosystem problems of ecosystem, and I will focus more on different concept of bifurcation, application of those concepts to understand certain biological phenomena at cellular level. So before we jump into those discussions, let us just try to recapitulate what is bifurcation. So if you remember, we are trying to understand the dynamics of a system in terms of one or more ODEs, and in these ODEs you will have dependent variables and independent variable. The independent variable is time, and apart from that there will be parameters which remain constant with respect to time. And in a system there can be a situation where the qualitative behavior of the system changes as we change the numerical value of the parameter. And if you remember, by qualitative behavior we mean that number of possible steady state in the system and stability of those type of steady states. So there can be situation where if I change the parameter value, number of possible steady state in the system changes, or the stability of the steady state changes, or both of them. In all these three cases, we will say the parameter is causing bifurcation in the system. So let us take an old example that we have discussed in the last module. I have a system represented by this set of two ODE, dx dt equal to m minus x square and dy dt is equal to minus y. So as we have discussed earlier, if I take m as a parameter and try to check whether it will has any effect on or it will cause bifurcation in the system or not, I can see that when m is less than 0, there is no real steady state, so we are not much bothered about that. When m equal to 0, you have one steady state, that is 0, 0, and I have drawn here the phase plane. So the phase plane is here and you have seen it earlier. So you have this 0, 0, the yellow dot, which is a saddle node type. Here on this side, trajectories are moving away, whereas from this side the trajectories are collapsing. Now if I change m and make it bigger than 0, there is a positive values, then I will have two steady state, minus root m and 0 and plus root m 0. In the phase plane you can see them, these two are the steady state, possible steady state when m is greater than 0. And one of them, this one, is a saddle node which is obviously unstable, whereas the other one is actually a sink, stable node. So you can see, if you remember what is happening here, as I am changing the parameter m, I have multiple different number of steady states and their stabilities are also changing. So I will say this system has bifurcation with respect to the parameter m. Now let us try to use these concepts on a biological process. Let us imagine there is a protein P whose expression is controlled by an external signal A. It may be like that, you have a plasmid, you have introduced a plasmid inside a bacteria which is a lac operon, and you can induce the expression of that gene under that lac operon by giving IPDG from outside. That can be your experimental system. So that type of system happens naturally in normal cells also. So you have a signal A which controls expression of a protein P, and there is another issue here. P also controls its own expression. So it's just like a positive feedback. I will not go in details about how to write the ODE for this system. I have written down the ODE and I will not, for the time being, will not explore different issues why I have written that type of OD. Just look at the OD that I have written here to model the dynamics of P with respect to time, how P will change with time. dP dt, that is the rate of change of concentration of P. So that is the OD I am writing. dP dt is equal to 0.1 is a constant plus 10 into a square by 1 plus a square, that a is the signal coming, into p square divided by 1 plus p square. P is the protein itself, minus p. Obviously the minus p part is for degradation of the protein, you can easily assume. Now let us look into the behavior of this OD. Now when I say I want to understand the behavior of the OD, most of the time I want to understand the steady state behavior of the OD. Now in this case, P is a dependent variable, T obviously is an independent variable, and A is a parameter. So I want to see the behavior of steady states of P, or the behavior of P with respect to defined values of A. So I will use the concept of direction field. So I have drawn the direction field here. I hope you remember what is direction field. In direction field I have T in the horizontal axis and P, the concentration of the protein, in the vertical axis, and I have this whole plane I have divided in grid, and at each grid I have drawn an arrow. Here the arrow head has not been shown, just only the line has been shown, and the slope of that each line at each grid point is equal to the derivative of P with respect to time dP dt at that position. So in this way you can see this red line shown here is actually a steady state, and in this case I have taken A equal to 0.1. So when I take A equal to 0.1, the system has only one steady state. Here the way I have shown the red line, and it is horizontal, that's why. And looking at the direction field, you can easily see that this steady state is actually a stable steady state. The steady state value may be around 0.1 something, and it is stable because if I start from, suppose here, I collapse with time, follow this trajectory and collapse at the steady state. If I start from a lower value, then also I move up and reaches that steady state. So from both sides I am reaching towards the steady state, so obviously this is a stable steady state. But notice the value of P is very low, very close to 0, that is around 0.1. Now what I will do, I will change the value of A to a higher value. So let us take A equal to 0.7, and again I have drawn the direction field with T in the horizontal axis and P in the vertical axis. You can see here again I have only one steady state, that is shown by this red horizontal line, and the value is near 3. So now the steady state has increased its value, it is at a higher value of P. And in this situation also, if you look at the direction field, you can easily understand that this steady state near 3 is also a stable steady state, because if I start at a lower value, suppose at t equal to 0, I am starting at this position, then the trajectories will be like this and ultimately collapsing at the steady state. If I start at a higher value from the steady state, again I come down along this trajectory and collapse at the steady state. So from both the direction I am collapsing, I am moving with respect to, when I am moving with time, I am moving towards the steady state. So this is a stable steady state. So what I have observed in these two direction field, that for a lower value of A, that is the input signal, lower value of input signal, I have a stable steady state whose value is lower, around 0.1 or something, whereas when I increase A to a higher value like 0.7, I have another steady state which is stable, but its value is higher, around 3. Now let us take a value in between for A, and what I have drawn here is A equal to 0.5. Now notice the direction field. Now we have 3 steady states. Here near 1.5, another one is here around 0.5, another one around 0.1. So I have now, as I have changed the value of A to 0.5, I have all of a sudden not one steady state but three steady states. Look at the stability. Let us follow the direction field to understand the stability. If I start from this higher value, suppose 1.7 or 1.8, the direction field arrow says that I will move along this trajectory, come down and collapse at this steady state near 1.5. If I start slightly lower value, I will follow this trajectory and move up and eventually with time asymptotically reach the steady state. So obviously this steady state is a stable steady state. So the steady state near 1.5 is a stable steady state. What about the middle one near 0.5? If I start near it but not at that steady state, the trajectories are telling me that I am moving away and going towards the higher steady states. If I start slightly lower, then the trajectory I am following is going down. So that means in this steady state, if I slightly deviate from this steady state, I diverge out. So obviously, this is an unstable steady state. What happens for the lower one? Let us clean a bit to understand what is happening. The lowest steady state, which is around 0.1, the trajectories are such that if I start from a higher value, actually I will collapse. If I start from a lower value, I will again collapse to that steady state. So that means this lower steady state, the third steady state in this system, is also stable because from both the side with time I am moving towards that steady state. So what I have observed in this three direction field, when I have a lower value of A at 0.1, the signal is 0.1, I have only one steady state which is stable. When I move to a higher value of 0.5, A equal to 0.5, the external signal equal to 0.5, I have three steady state, out of which two are stable, one is unstable, the middle one is unstable. Then if we further increase A to 0.7, then again 3 disappears and only one steady state comes, and that has a higher value and it is a stable steady state. So I can compile all these data and draw something which we call bifurcation plot. And that's what I have done here. So if you remember, in the bifurcation plot, usually we put the parameter which is causing bifurcation in the horizontal axis and the steady state value. Remember, the steady state value of the dependent variable in the vertical axis. So we have the steady state value of P in the vertical axis here and the activator A or the signal A in the horizontal axis. And I can show mathematically, I am going to draw this one, and from the previous data also you can understand easily that when activator A, the signal A is at lower value, say up to this range, I have only one steady state which is stable and has a lower value, this region, this blue region. Whereas when I have a higher value, I have for like this one, 0.6 to 0.7, I have this one. The steady state is stable or its value is higher. In between, suppose when I take 0.5 or 5.55, I have three steady state, 1, 2, 3 for 0.5, we checked. This one is unstable. This one is stable. And this one is also stable. Let us take another value here around 0.55. Then also I have a stable steady state here. I have an unstable steady state here. You can check by calculating the different direction field. And I have another stable steady state here. So what we are observing, if I look into the whole map here, if I clean a bit, I can see that depending upon the value of external signal or activator A, I have three regimes. One regime is here. When the system in this regime, I have only one steady state. The system has only one steady state. P has only one steady state, and that is the lower value. Whereas I have another regime here when A is bigger than this value, I have one steady state only for P, and that is the higher value. In between these two, I have a region where depending on for any value of A, I will have 3 steady state, out of which 2 will be stable, 1 will be unstable. So now, how does this can affect the behavior of cells in a population? Let us try to understand that. See, when suppose I am growing cells in a flask, all these cells are isogenic. That means these all cells are genetically identical. But that doesn't mean at any moment of time all cells will have same amount of a particular protein. There will be slight variation. So if I can measure the amount of protein P at a particular time point in a ensemble of cell, in a population of cells, suppose I have 1 million cell, and if I can measure amount of P in each of these cell, then I will not get exactly same value in each cell, rather I will get something like a distribution. Suppose this is the protein concentration, and I will have a histogram where majority of them has this mu value, and some of them will have higher value of the protein, and some of them will have the lower value of the protein. So I always get a distribution of protein expressed in a set of cell, a population of cell. And that's what you observe when you do flow cytometry experiment. You don't get a single value of reading for a particular protein in a population of cell, rather you get a histogram which looks like a bell-shaped histogram. So now imagine, let me clean this part a bit, it will be easier for us to understand. So suppose I am giving a signal A to a population of cell, and the value of that signal is suppose at 0.4 here. So then the steady state value of P on an average will be this one, because there is only one possible steady state. Now as I said, in a population of cell, all cell will not have the same amount of protein, but rather I will have a distribution. So I can consider this steady state value that is shown in the bifurcation plot is the mean value, and the amount of protein in the population will be distributed around this. So I can draw a histogram like this. So that's what is drawn here. So this is the mean value of the protein. Now when you increase the value of the signal, suppose from external agent I am adding extra signal and make this signal A to 0.7, then the new steady state will come after some time, and all cell will move toward this particular steady state. So at this steady state, the average value of protein in the population, in the cell population, will be this value. But again, I will have a distribution. So all cell will not have the same value, but rather you have a distribution around this. So that's what we get here. So the mean value is this one, and you have a distribution around this. So when I have a low signal, I have a population distribution like this. When I have a high signal, I have a population distribution like this one. In between, what will happen? If I give a signal of 0.5, I have two possible steady state. So suppose initially cells were here, and the signal I am giving from outside was 0.4, and then I have added few drops of the signal from outside, and the signal increased to 0.5 now. Now if I allow this system to go to a steady state, I will have two possible steady state. One is here, the other one is here. You have an unstable steady state here, but as the steady state is unstable, no cell will go there. So based on the initial value of protein present in the cell, some of the cell will move to this steady state, some of the cells will move to this steady state, no one will move to this unstable steady state. So I will have a population whose average value will be this one, lower steady state, stable steady state, and there will be another subpopulation which will have average value of protein P in them at this upper stable steady state. So what I will observe? if I measure protein P in each of the cell and then draw a histogram, now I will have histogram like this with two peaks. This lower peak is the average value coming from this lower steady state. So this is coming from the lower steady state, and this peak, this is coming from the upper steady state, and both of them are stable. No cell will reach this intermediate steady state which is unstable. So what is happening here? If you follow this population behavior data, when the signal is low, I have a population of cell where all the cell amount of protein in these cells are distributed around a single mean. And as I increase the amount of A or signal A situation comes when from one peak I get two peak. So I have now two subpopulation. So the whole population is now divided into two subpopulation. And again, if I increase further towards the high value of 0.7, two population will merge and a new population will arise, which is homogeneous population having a higher value of P. So I have initially like this and then this one. So this type of behavior is called bimodal distribution. Initially I have unimodal population distribution. In between, when I have two peaks, this is called bimodal distribution. This is called bimodal because you can easily see there are two modes, there are two peaks. So what is happening here if I brief? If I have a system in which, depending upon external signal, I have bistability, because if you look at the bifurcation plot here, I have one stable steady state here, another stable steady state here. So this system is called bistable. So it has two steady states which are stable. So if I have bistability in the system, then if I start with a homogeneous population initially, and as I keep on changing the signal, I may get from a homogeneous population, I may get two subpopulation with a bimodal distribution. And again, if I keep on increasing the signal, again I may get a homogeneous population with higher expression. So if from unimodal I get bimodal, then again I get unimodal distribution. So this example shows that a simple feedback circuit, the ODE which I have written, is essentially nothing but a simple feedback circuit for protein expression P that has bistability and that give rise to a bimodal population distribution of protein. Now let us try to use it further. We know that cells differentiate. You may have a multipotent stem cell. From that multipotent stem cell, a signal comes and the stem cells differentiate into another type of cell. But all cell does not differentiate. If all cell differentiate completely into a new type of cell, then we will not get this body organization during our embryonic development. Some of the cell will differentiate into type A, some of the cell will differentiate into type B, something like that. So this type of phenomena can be explained by the bifurcation and bistability and bimodality that we discussed just now. Let us imagine initially I have a homogeneous population expressing this yellow protein and this red colored protein in triangle shape. It is a homogeneous population. Now these proteins are controlled by a network or circuit which shows suppose bistability or multistability. So if it is showing bistability, then as we give signal, increase the signal, a situation will come where from a homogeneous population, two subpopulation will appear. One having higher expression of the protein, another having lower expression of the protein. So in this case you can see for this cell, for example, the red proteins are higher, whereas for this cell the yellow protein are higher but red protein are lower. So what is happening from a homogeneous population where most of the cell has almost close number of red and yellow protein, as the signal is given and as the system has bistability for both the red and yellow protein, I have two populations, subpopulation now, one having higher red protein and another having higher yellow protein. So imagine this yellow protein and the red protein controls differentiation of the cell. So suppose maybe the red protein pushes the cell towards neuronal cell type, whereas the red one, the yellow one pushes it to a different cell type, type B. So what will happen? These cells which is expressing more of red now, which is a subpopulation expressing more red now, will become one type of cell. Whereas the other cells subpopulation which is expressing more yellow will become another type of cell. So from a homogeneous population, I get two subpopulation and their protein expression behavior are different, and ultimately they move into different path of differentiation. This type of observation we have made not just theoretically, people have studied this type of behavior for different molecules like nano, which are key transcription factor involved in differentiation of mammalian system, but even in a human. So in those cases, we have observed that the network which controls the expression of this protein are bistable, and at a particular signal, with a proper signal, this bistability can give rise to two subpopulation of cell having a bimodal distribution. Now this idea can be extended further to understand the embryonic development. We know that we all start from a lump of mass, a lump of cell where all cells are identical. Then these cells differentiate in different paths and we get this body organization. So it is a long-standing problem that how this differentiation happen and bifurcation happens, that different types of cells originate from one single cell type. What is the dynamical reason behind it? Long back, Waddington proposed something called Waddington's epigenetic landscape. He used the metaphor of valleys and rivers. So you can imagine, suppose the whole space has valleys and hills. So initially the embryo may be somewhere here. Then as the time progresses, different input signal comes, the cells move in this trajectory, just like they move on a phase plane, and they have only one stable steady state here. Then as time passes further and input changes, now the system may have two stable steady state, one here and one here. So some of the cell in the population will move towards this steady state and the other will move to the other steady state, obviously depending upon their initial condition in the phase plane. Suppose they are farther down in time, and depending upon the input signal, the system now can have four steady state, stable steady state here, here, here and here. All these four are stable steady state. So the cells which are here in this particular steady state now either will move towards this one or will move to this one based on their initial conditions, and cells present in the other steady state will either move to this steady state and this steady state. Each of these steady state, suppose, represent one particular type of cells. So using the concept of bifurcation, we can easily understand that from one single cell type, as the time progresses and the input signal changes, depending upon the input signal or the parameter value, the system goes through multi-stable or bistable situation. And as we go through bistable or multi-stable situations, cells will bifurcate and they will move into one of these stable steady state, and they will differentiate into different cell types. Let us look into another interesting thing that can happen in cell biology due to bifurcation. I have drawn the same bifurcation plot here that we discussed some time back. Suppose initially I am treating the cells with a signal 0.3. Then if I allow the system to reach steady state, the steady state value of P will reach here. Then I add some more amount of the signal A or activator A, and suppose the total concentration becomes this one, 0.55. So now, if I allow the system to reach steady state by waiting for some time, then cells will have three possibilities. The system will have three possibilities. Three steady state. Obviously, the middle one is unstable, the system will not move there, but the system has the option to either stay in this lower value or to jump into the higher one. Using common sense, and if you look into the direction field, or in case of a more than one ODE, you can easily understand that as initially the system is in the lower steady state, this lower blue line, it will try to stay at this lower steady state. It will not jump into the higher one. This will happen till I reach this point near point 6. So in this whole range up to this value of A up near point 6, the system will always remain on this blue line, although for some value of A, it has a chance to go to higher value, but it will not go there because on the phase plane or direction field it is far away. So the system is already in the lower steady state, so it will stay in the lower steady state. If I increase the value of A further, there is no other option, but the system will jump to the upper steady state in the green line. So if I keep on increasing the value of A towards 0.7, the system will jump along this green line and may reach to this highest value. So the trajectory is, initially it will move like this and then it will move like this. Now reverse the process. Suppose already the cells are at this higher steady state and the signal is also at 0.7. I reduce the signal, I change the media, or I do something so that the amount of A in the culture media decreases, so that the cells will see less amount of the activator and allow the system again to reach steady state. So where it will go? Obviously it will follow this green line and drop somewhere here. Then if I reduce it further, it will go somewhere here. Now if I reduce A further, then suppose I reduce A to this value at 0.55. So at 0.55, again the system has two options. Either it can stay on this green line, which is a stable steady state, or it can also jump to this lower one, which is also a steady state on the blue line. So in this 0.55, again the system has two steady state which are stable. One is the higher one is the lower one. The question is, will it divide or it will jump to lower one? In fact, if you look into the direction field or phase plane plot for this type of problem, you can easily understand that as the system is already on a stable steady state, it will not jump into the lower blue line, rather it will remain in this green line. So what will happen? When I am decreasing these values of the activator A from higher value, it will follow the green line up to this, and then it will have no option, so it will follow this path, the lower blue line. So if I draw it with clarity, when I increase the value of A up to this value near 0.6, from 0, from lower value to 0.6, the system will stay in the blues or the lower steady state, and then it will follow this path. So my path is like this and then jump and increase. When I start from a higher value of the steady state and decrease the signal or activator A, it will follow this path and then it will jump to this lower one, and if I decrease A further, it will go follow the blue path. So you can easily see here the path followed by the system when I am increasing A from lower value to higher value is different from the path followed by the system when it is coming back from a higher value of the signal A to a lower value of signal A. This typical behavior of two different paths is called hysteresis. Hysteresis you may have seen in case of polymers in many other material science discussion. They are also the paths of change in physical properties of those material are different depending upon how you are applying the force. So here also, as I increase the input signal, the system follow one steady state path, and when I decrease the signal from a higher value, it follows another path. Now, let us look into an concrete example of this in an experimental system. And I have taken this from a journal paper in Nature. So what they have taken bacterial cell and have introduced a plasmid system there which will express GFP, and the GFP is under the control of a network of positive feedbacks. I will not go in detail of that. In essence, the GFP is controlled primarily by a lac operon. So from outside I can give IPTG or TMG to induce the whole system, and GFP will be expressed, and the figure here shows GFP expressed in bacteria. And if you remember, all cells will not express the same amount of GFP based on the same induction value, because I will always have some amount of difference in the expression of protein from cell to cell. So what they did a very simple experiment. When the constant they used initially, they took cells which are uninduced. So those uninduced cell has very low expression of GFP, and you get very low value of GFP intensity. So then they keep on increasing the amount of TMG, that is the inducer, in this flask. So initially they have added this amount of TMG, that may be 3 micromolar, and then they measure the GFP intensity in a population of cell. So here the data is shown as dots, each dot represents one cell. You can see on an average the value of intensity is somewhere here. Now, after some time, they increase the amount of TMG in the system to 6 micromolar and allow the system again to reach a steady state. Then I get these data. Still, the average value, on an average, the value of steady state value of the GFP is at a lower steady state value. They kept on doing it. After 10 micromolar, you can see they are getting two populations. Population 1 here, second population here. That means the system is reaching the zone where I have bistability. One stable steady state is here, another stable steady state is here. As they keep on increasing amount of TMG, you can see two populations distinctly arise, and then at this arrow, after that, when they increase TMG further, we have only one population here which has a higher steady state value. So what is happening? They took uninduced cell and they started adding TMG to reach a particular value of TMG concentration, and allowed these cells to reach a steady state and measure the intensity of fluorescence of GFP in each of these cells and plotted the data. And you can see initially the system remain at this lower steady state, and then bistability arise and majority of the cells are in the lower one, then slowly they move to the higher one. Now what will happen if I take these cells which are already induced with high amount of TMG? So already I have increased TMG in the system. So already I have a population which is induced with high amount of TMG, around 30 micromolar. Now if I start reducing the amount of TMG in the system, that is the input signal I am reducing in the system, what type of profile I will see? That's the experiment they started doing. So here you have the starting point t equal to 0, where cells are induced with high amount of TMG, around 30 micromolar. So they have high amount of expression of GFP. As they decrease the amount of TMG, you can see the cells are still remaining at the higher value. They are not dropping in any lower value of steady state value of GFP, although they have reached the region of this region where bistability is possible. So as they started with a higher value of GFP expression, higher steady state of GFP, they are staying there till this value of around 4 micromolar of TMG, where the lower steady state GFP cells appears. And here at the end, I have only one population having the lower expression of GFP. So you can easily see, when I am increasing TMG, my path is this one. When I am decreasing TMG, my path is this one. So these two paths are distinctly different. That means this system has hysteresis. The cells which were induced slowly from lower level to higher level and eventually have seen large amount of TMG, they remember the molecular information, and when you try to reduce the amount of TMG, they don't fall back to the lower steady state value, but they remain at the higher steady state value of GFP for prolonged period, and then eventually when they go out of the bistable region, they drop down to the lower steady state value. So here I have a very simple example by which bistability give rise to hysteresis, and hysteresis is nothing in this case but a form of molecular memory. Let us jot down what we have discussed in this module. First, we have discussed that bifurcation can happen in many systems in biology. I have focused mostly in case of cellular system. For example, when a particular protein is controlled by its positive feedback, or I have hysteresis in the system. Now bifurcation with bistability and multistability can happen. In case of bistability, you have two stable steady state. In case of multistability, you have multiple stable steady state. Now this type of bistable or multiple stable system can give rise to cellular heterogeneity. We have discussed that. And this type of cellular heterogeneity eventually may give rise to a bimodal or multimodal cell population, depending upon the number of possible steady state available to the cells. And finally, we have discussed that bifurcation can give rise to hysteresis, and hysteresis in a way give rise to molecular memory in cells. That's all for this module. See you again.